Imagine, if you will, a sprawling subterranean city, just thousands of individuals bustling about in the pitch black.
Right, completely underground.
Yeah, and they're gathering food, they're continuously excavating these complex tunnel systems, and fiercely defending their borders against intruders.
Very fiercely.
But in this massive, highly organized society, only one single female is allowed to have children.
It's a complete, absolute monopoly on reproduction. I mean, it is a social structure that defies almost everything we expect from mammalian biology, and yet it exists right beneath our feet.
Welcome to our deep dive into the source material. Today, our mission is to unpack a truly wild discovery from a July 2026 paper in the journal Nature.
Yeah, focusing on the naked mole rat.
Exactly. And we've known for decades that their societies operate much like a beehive or, an ant colony.
Which is a phenomenon called eusociality.
Right, and that's incredibly rare in mammals. But the mystery has always been, well, how does one female physically stop dozens of others from reproducing? Like without a police force or physical barricades?
That is a million-dollar question.
So today, we're breaking down the exact chemical weapon the queen uses to suppress her colony, how she deploys it, and the biological cascade it triggers in the brains of her subordinates.
It's just an incredible piece of research because it finally reveals this ultimate invisible tether. I mean, the researchers haven't just identified a concept, they've isolated this specific molecule.
Which is wild.
It really is. They tracked its path through the olfactory system, and they mapped out how it fundamentally alters the endocrine system of an entire society.
So let's start at the very foundation here. To understand how a smell can control a mammalian society, we have to look at how naked mole rats use their noses in the first place. Right?
Yeah, absolutely.
Because these animals live entirely underground, vision is just completely useless to them, and their society is strictly tiered.
Right, you have the smaller, low-ranking individuals who are essentially the workers. They maintain the tunnels and find food.
And then you have the larger, high-ranking members who act as the soldiers.
Exactly. And because they can't see, the researchers needed to confirm that olfaction, their sense of smell, is the primary way they navigate this complex hierarchy and identify threats.
Makes sense.
So they started with a very straightforward recognition test. They took naked mole rats and placed them in observation tubes, pairing them either with familiar mates from their own colony or with complete strangers.
And the behavioral difference was stark, right? I mean, they spent significantly more time intensely sniffing the foreigners.
Yes, much longer.
But I always wonder with studies like this, how do you prove that smell is the absolute driver? Because if I meet a stranger, I might look at them longer, but I'm also listening to them, watching their body language.
Right. There are other variables.
So how did they isolate just the sense of smell?
Well, they essentially had to temporarily turn off the mole rats' noses.
Oh, wow. How do you even do that?
So to do this, they used a specific drug called methimazole. When injected, it causes rapid targeted damage to the olfactory sensory neurons inside the nasal cavity.
Just completely wipes them out.
Pretty much. Within about 72 hours, it specifically destroys the mature OMP-positive neurons and the immature SDMM1-positive neurons. So they essentially have no sense of smell.
Wait, let me make sure I'm wrapping my head around this. You're temporarily cutting the wires to the colony's main security camera system, basically.
That's a great way to put it, yeah.
But if you take away my primary sense, I don't just go about my day normally. I'd be totally disoriented. Wouldn't destroying their sense of smell just make them, I don't know, sluggish or sick or entirely passive?
That is exactly the right question to ask, and the researchers tracked this carefully. The animals did experience a transient drop in body mass.
Because they couldn't smell their food.
Exactly, they had trouble locating it. But that resolved within about 3 weeks. And what's crucial is that they did not just curl up in a ball and become passive.
Really? They were still active?
Very much so. To test their aggression, the researchers set up a neutral arena and introduced high-ranking soldier mole rats from different colonies to each other.
Okay, setting up a little fight club.
Basically. And when two healthy soldiers met, it was immediate overt aggression. We're talking head-to-head shoving, biting, chasing.
Wow.
But when two smell-deprived soldiers met, the aggression plummeted to almost zero.
But hold on, did they lose the ability to fight or just the trigger to fight?
That is the brilliant part of the experiment. When the researchers mixed the groups, putting a healthy soldier in the arena with a smell-deprived one, the healthy mole rat would immediately attack.
Because it could smell the intruder.
Right. And the smell-deprived mole rat would actively and fiercely fight back.
That is fascinating. So they still have the physical capability, and the instinct to defend themselves is fully intact.
Exactly.
They just completely lost the ability to initiate the conflict because they couldn't smell that the animal in front of them was a foreigner.
You nailed it. And even more interestingly, despite losing their ability to read these chemical cues, the social hierarchy of the colony didn't collapse.
Oh.
Yeah, their rank remained completely stable. This proves that while rank is maintained by other things like body mass and vocal cues, the trigger for actually defending the colony is purely chemical.
Okay, so let's pause here for a quick recap for you listening, just to summarize what we've covered so far. First, these eusocial mammals act a lot like bees.
Right.
Second, naked mole rats use smell to identify strangers. And third, destroying their olfactory neurons stops them from initiating fights, but they can still defend themselves.
Perfect summary.
And I have a question for you to mull over. If you suddenly lost your ability to read basic social cues, how would your daily interactions change? It really makes you think about how much we rely on invisible signals.
It demonstrates just how heavily these animals rely on chemical communication, which naturally brings us to the core of the mystery.
Right, the transition from defense to reproduction.
Exactly. Because if smell dictates who they attack, does it also dictate who gets to reproduce?
If the colony's defense mechanism is entirely scent-based, it definitely stands to reason the queen's control mechanism might be too. But trying to find one specific chemical in a whole subterranean colony sounds like a nightmare.
It was a massive undertaking.
How do you even begin to hunt for the queen's specific scent?
Well, the researchers analyzed 771 samples across 351 mole rats. They were taking samples from all ranks, ages, and reproductive states.
That is a lot of sniffing.
Yeah, and they detected 240 distinct volatile chemical compounds.
240 compounds. That's just a wall of noise. How did they figure out which one was the weapon?
They used a statistical method called a principal component analysis, or PCA, to group this massive data set and look for outliers.
Okay.
They were looking for something completely unique, and out of those 240 chemicals, one single compound stood out entirely on its own.
And what was it?
It's called isopropyl myristate, or IPM.
IPM. So what makes this one molecule so special?
Well, when they looked at the distribution, IPM was highly enriched specifically in the queens. It was practically non-existent in the non-breeding males and females. They did find trace amounts on newborn pups, but that's likely just from physical contact with the mother.
Right, from rubbing against her.
Exactly. They swabbed different body sites and found the highest concentrations in the queen's vaginal, anal, and salivary secretions.
But not everywhere.
No, it was almost absent from her urine and milk.
So it's not just a general waste product. It's a very targeted secretion.
Precisely. And the timing of its release is the real smoking gun here. IPM levels don't just stay static.
Oh, they change.
Yeah, they skyrocket, specifically during the queen's ovulation phase. And then they drop significantly when she becomes pregnant or starts lactating.
Wow, so it is a massive chemical broadcast of her reproductive readiness.
Exactly.
I want to talk about the physical nature of this chemical, too, because it explains so much about how the queen behaves. In the source material, it notes that IPM is a long-chain ester with very low volatility.
Right. That's a key physical property.
So to visualize this, think about perfumes. You have those, cheap aerosol body sprays that instantly fill a room, but then they fade in 5 minutes because they're highly volatile.
Yeah, they evaporate quickly.
But IPM is not that. It's more like a heavy, expensive, oil-based perfume. It lingers for a long time, but it doesn't really travel very far in the air on its own.
And that property is vital. Because it's heavy and low volatility, it can't just passively waft through miles of underground tunnels.
Exactly. Which perfectly explains a behavioral quirk of the queen that researchers have observed for years. She is incredibly hyperactive.
But she never stops moving.
Right. She constantly patrols the colony tunnels. She's shoving past workers, constantly moving. She isn't just micromanaging her workforce. She is acting as a physical diffuser.
Yes.
She has to actively rub this heavy oil onto the walls of the tunnels and onto the bodies of her subordinates to ensure every single member of the colony is exposed to it.
She is essentially painting the walls with her authority.
Yeah.
It's a brilliant way to chemically dominate a massive physical space.
Think about a time you walked into a physical space and instantly recognized a specific lingering scent, like maybe an old bookshop or a grandparent's house that just has a smell baked into the walls.
Oh, for sure.
So time for our second recap. Here is your bullet summary. 1. Researchers isolated 240 compounds and found IPM is unique to queens.
Yep.
2, IPM peaks during ovulation. And 3, it's a low volatility chemical which forces the queen to actively patrol to spread it.
That covers it.
And here's a question for you. Knowing how a physical space can retain a scent for days, how does that change how you view the queen's constant patrol of her underground tunnels? She's basically making sure the house always smells like her.
But having the chemical and knowing how she spreads it is really only half the equation here.
Yeah.
The researchers then had to figure out what smelling IPM actually does to the brain of a subordinate mole rat.
Right, because if I smell my grandmother's perfume, I might feel nostalgic, but it definitely doesn't chemically sterilize me.
Let's hope not.
Right, so what is happening in the mole rat's brain when they smell this?
The neurobiology here is spectacular. To see the brain's reaction, they use 2 different imaging techniques. First, tissue clearing, combined with FOS immunostaining.
And what does that do?
FOS is a marker that lights up in neurons that have just recently fired. So when they exposed non-breeding mole rats to IPM for 90 minutes and then looked at their brains, they saw a significantly higher density of these glowing active cells in the olfactory bulb.
Compared to the ones that weren't exposed, I assume?
Exactly.
But looking at glowing cells after the fact is just a snapshot. The source material notes they wanted to watch the brain react in real time, right?
They did. So they turned to functional ultrasound imaging, or FUSI. It's a non-invasive way to measure microscopic changes in cerebral blood volume.
Okay, cerebral blood volume.
Right. The underlying principle is simple. When a specific brain region works harder, those active neurons need more oxygen, so more blood flows to that area.
Got it.
And the ultrasound can track those blood flow changes with incredibly high resolution as they happen.
So they essentially waft this heavy IPM scent under the nose of a worker mole rat while monitoring its brain. What lights up?
It was a dramatic response.
Yeah.
They saw massive stimulus-locked blood flow increases, not just in the initial olfactory bulb, but deep within the brain.
Like where?
Specifically, the piriform cortex, the geniculate nuclei, and areas intimately associated with the amygdala.
The amygdala. Wait, that's the emotion and fear center of the brain, isn't it?
It is. So this scent isn't just being processed as some neutral environmental odor. It's triggering a profound emotional or survival-based neurological response.
Wow.
And one really fascinating physiological quirk they noticed was that the brain response was highly lateralized.
Meaning what? It was on one side?
Yeah, mostly lit up on one side of the brain at a time.
Hmm.
This is tied to the nasal cycle, which is a phenomenon where mammals naturally alternate which nostril is dominant, drawing in the most air at any given time to optimize how we process smells.
That is so cool. But wait, I have to jump in here with a detail from the paper that genuinely threw me.
Oh, what's that?
The researchers note that this chemical, isopropyl myristate, is actually found in human breath and in the areolar secretions of lactating human mothers.
Yes, it is.
Which immediately made me wonder, does this mind control chemical affect us? Like, if someone breathes on me, are my hormones changing?
It's a very natural question, and it brings up a fascinating point about evolutionary biology. The researchers actually ran a human trial to test this exact thing.
Oh, thank God.
They screened 766 different human odorant receptor variants. And the result was that humans essentially perceive IPM as completely odorless.
Oh, really?
Yeah, our receptors showed only very weak, non-dose-dependent responses. So while our bodies naturally produce this chemical, we do not have the hardware to react to it anymore.
Okay, so I'm safe.
You're safe. Naked mole rats, on the other hand, have a vastly expanded repertoire of specific odorant receptors. Over evolutionary time, they have developed a highly specialized communication channel explicitly tuned to this one molecule.
Okay, let's do a quick recap of Section 3. First, IPM physically activates the olfactory bulb and the amygdala, which is the fear center.
Right.
Second, functional ultrasound imaging, or FUSI, tracks this in real time via blood flow. And third, humans have the chemical but lack the receptors to react to it.
Good summary.
And a question for you listening, knowing that an odorless chemical can dramatically alter a mammal's brain blood flow, how much of our own behavior might be influenced by things we can't even consciously perceive?
It certainly highlights the invisible chemical environment we all live in. But for the mole rat, that brain signal is just the beginning of the cascade.
Right, because feeling fear is one thing.
Exactly. We still have to trace how a triggered amygdala translates into physical sterilization.
Yes, let's follow the chain of command inside the body. We've gone from the queen's scent into the tunnels up the nose and into the brain. How does the body actually shut down reproduction?
So first, they looked at the behavioral response to the scent. They placed the mole rats in a T maze.
Like a tunnel that forks left and right?
Exactly. One side contained the IPM scent, the other was blank. Now, the lowest-ranking workers, the ones who were furthest from ever reproducing, they didn't really care. They explored both sides equally.
Okay, what about the higher ranks?
The high-ranking non-breeders, the beta females, who are essentially next in line for the throne, they actively avoided the IPM-scented chambers. It physically repelled them.
And I imagine if they tested the mole rats who had their sense of smell temporarily destroyed with that drug, methimazole, they wouldn't avoid it, right?
Correct. The smell-deprived ones walked right in, which proves the avoidance is purely driven by the scent.
Wow.
So the individuals with the highest reproductive potential are the most terrified of the queen's specific odor.
But fear alone doesn't sterilize you. There has to be a physiological mechanism backing this up.
There is, and it's a profound hormonal shift. When these non-breeding females are exposed to IPM, it literally alters their endocrine system. Specifically, it significantly raises their circulating levels of a hormone called prolactin, while simultaneously suppressing their levels of progesterone.
Okay, let me think through this. Prolactin-like lactation. In humans and almost all other mammals, a massive surge in prolactin happens during nursing to stimulate milk production, right?
Yes, it does.
And biologically, doesn't high prolactin act as a natural contraceptive? It's called lactational infertility, keeping a mother from getting pregnant again while she's nursing a newborn.
You're exactly right.
Wait, is the queen tricking their bodies into thinking they are nursing?
That is exactly what is happening. The queen's scent is hacking the subordinate's endocrine systems by hijacking an ancient mammalian hormone pathway.
That is wild.
It is a brilliant evolutionary shortcut. By chemically forcing an elevation in prolactin, she inhibits the release of gonadotropin-releasing hormone. This completely shuts down ovarian activation, which keeps progesterone levels practically at zero.
It's biological sabotage. She's locking the beta females in a state of suspended animation, chemically mimicking a pregnancy or nursing state, so their bodies physically cannot prepare for a real pregnancy.
It is the ultimate biological repurposing, taking a system designed for maternal care and weaponizing it for social control.
Unbelievable. Okay, time for another recap for you. So, bullet 1, high-ranking females actively avoid IPM. Bullet 2, smelling IPM raises prolactin and lowers progesterone. And 3, this mimics lactational infertility, hormonally blocking reproduction.
Spot on.
And here's a thought for you. The queen's scent exploits an ancient pre-existing mammalian pathway to control her rivals. Can you think of other systems in nature that have been, hacked for a completely different purpose?
It really makes you appreciate the creativity of evolution, which leads us to the final and, frankly, most dramatic phase of the researchers' work, putting this chemical to the ultimate test.
Right, because it's one thing to watch hormone levels fluctuate in a controlled lab maze. But can this one single chemical hold an entire complex society together on its own?
To find out, the researchers first ran an isolation experiment. They took male and female non-breeders out of their colony, completely away from the queen, and put them in private opposite-sex pairs.
Okay, setting the mood.
Exactly. And the control group, who received normal, clean bedding, acted exactly as you'd expect. Freed from the queen's suppression, they quickly mated. The females gained 20 to 30 grams, and 5 out of the 6 control females became pregnant.
Nature taking its course. But what about the experimental group?
For the other isolated pairs, the researchers added a tiny amount, just 500 microliters of synthetic IPM to their bedding every single day.
And what happened?
The results were absolute. 0 pregnancies out of 7 pairs.
0.
0. Not only did they fail to mate, but their external reproductive organs never even fully matured. The synthetic chemical, completely isolated from the physical presence of a queen, was enough to completely halt their reproductive development.
That is staggering. But the source material notes they went even further with a full colony test, right? I like to think of this as the Wizard of Oz simulation.
It really was.
They took a fully intact colony of 19 mole rats, and they simply removed the queen.
Now, under normal circumstances, removing a naked mole rat queen is a catalyst for absolute chaos.
Oh, I bet.
Yeah, without her constant suppressing presence, the prolactin levels in the beta females start to plummet. Their progesterone spikes.
Preparing them to breed.
Right. And this triggers a bloody, chaotic, and often lethal war for succession as the females fight to become the new queen.
But in this specific colony, the researchers acted as the man behind the curtain. Every single day, they secretly added synthetic IPM to the colony's bedding. What happened?
For 12 solid weeks, there was absolute peace.
You're kidding.
No, the hormonal data perfectly matched the behavioral observation. Prolactin stayed artificially high. Progesterone stayed suppressed. There was no aggression, no dominance contest, no succession war whatsoever.
Totally normal.
The entire colony functioned smoothly, suppressed entirely by the chemical ghost of a queen.
12 weeks of peace, maintained solely by a bottle of synthetic scent. But eventually they had to pull the curtain back, right?
They did. They finally stopped adding the IPM, and the illusion shattered almost immediately.
How fast?
Within a month of the IPM withdrawal, the prolactin levels in the colony dropped. By week 16, severe aggression spiked among the females. A lethal attack actually occurred, leaving one female dead.
Oh, wow.
And by week 19, the surviving victor rapidly gained weight, her progesterone levels skyrocketed, she became pregnant, and she officially took her place as the new queen.
It's incredible how fast the social fabric tears when that invisible tether is cut.
It really is.
And this mechanism isn't just a fluke of this one specific colony, right?
Far from it. To put a bow on the evolutionary context, the researchers tested other species too. They detected IPM in the breeding females of 4 other eusocial mole rat species, like the Damaraland mole rat.
Okay, so it's common in the eusocial one.
Yes, but it was completely absent in solitary mole rats, like the Cape mole rat, who live alone and just don't have these complex societies.
Which proves this isn't just a quirk of mole rat biology in general. This is a highly specific convergent evolutionary adaptation designed explicitly for social control in extreme hierarchies.
Exactly.
So final recap time. First, synthetic IPM entirely prevents isolated pairs from mating. Second, adding IPM to a queenless colony maintains peace and suppresses reproduction for months.
Yep.
And third, removing the IPM immediately triggers a violent succession war.
Perfectly summarized.
And here's a question for you. If a single molecule can maintain social order and prevent violence in a mammalian society, what does that say about the concept of free will in highly social animals?
It really bridges a massive gap in our understanding of biology. We've long known that insects like bees and ants use chemical caste systems. But finding such a profound single-molecule control mechanism in a mammal completely reshapes how we view the evolution of mammalian social structures.
So we started today by imagining a subterranean city where one ruler enforces total reproductive control through a scent. And we've seen exactly how she pulls it off.
Through IPM.
Yeah, a simple, heavy, long-chain, fatty acid, ester isopropyl myristate acts as a master key. It's a social hormone that physically links the physiology, the brains, and the behavior of an entire society together.
And it does so by hijacking the very hormones that normally foster maternal care, turning them into a tool of absolute suppression.
It is brilliantly terrifying. I want to leave you with a final, slightly provocative thought to mull over.
Oh, what's that?
Well, we talked about the fact that humans naturally produce this exact same chemical, IPM, in our breath and in mother's milk.
Right, even though we don't react to it.
Right, we don't consciously react to it now, and we lack the specific receptors the mole rats have. But if naked mole rats evolved this highly specific chemical language to dictate social hierarchy and reproductive status, is it possible that deep in our own evolutionary past, our ancient mammalian ancestors once communicated social hierarchy through these exact same invisible chemical signals? Wow. Have we simply forgotten how to read a language that our bodies are still quietly speaking?
That is a fascinating reminder that there are deeply complex chemical conversations happening all around us every second of every day, whether we have the receptors to tune into them or not.
Next time you walk into a room, just think about what invisible messages your nose might be missing. Thanks for taking this deep dive into the source material with us. Keep questioning the invisible world around you.